So the next bunch of slides have a series of animations. So what I would suggest doing, whoever's controlling the slides, is just to advance them all the way to the end, and I'm just going to talk for a minute on each one. So there's 3 case studies that we're using for driving our design, and actually we have more than 3 case studies, but I'm going to talk about 3. One of them is the prospect of, say, engineering a microbe for more efficient biofuel production. Okay, and what we mean by that at a very high level is that we don't want to produce alcohol from organisms, from sugars. This is a very, taking corn and producing ethanol is not what we're after. What we're after is taking much more complex inputs that might be just biomass from things like switchgrass or corn stover or whatever, and engineer an organism to produce chemical precursors to fuels that are much closer to the fuels that we actually want, closer to biodiesel or gasoline or so on. And here's the steps needed to do that. We have to, there's a lot of sequencing involved. From that, we need to annotate the genomes. We need to build these metabolic models I've been talking about, compare that to experimental data on that. We need to build regulatory models that control, so that the cell has sort of 2 types of networks, at least 2. A metabolic network that is really the chemistry, the energetics in the cell, the synthesis and so on, and then a regulatory network is sort of the control layer that sits on top of that. It tells the cell when to do what. Might also have signaling that it uses to coordinate with other organisms. So we want to reconstruct both the metabolic network and the regulatory network. We want to take kinetics data if we have it and fit that. We want to then predict what kinds of throughput we would get in certain pathways. If we, in this case, we want to produce, say, isoprene as a precursor for a biofuel. We want to engineer that organism so that it devotes most of its energy to producing isoprene. And based on this computational analysis, we would then make a set of predictions, a set of recommendations to people that are going to engineer the cell to delete certain genes, add certain genes, change the regulatory network actually to be optimized for this. So this is a type of a workflow needed to actually do that. And of course, KBase is building the infrastructure for us to support that kind of analysis. There's 2 more of these examples. I think in the interest of time, I think we should just skip through them, but just to know that there's similar workflows in, say, engineering plants to be better feedstocks for biofuels, say, or to be more resistant to salinity, or to be more effective in utilizing nutrients in the soil, or whatever you want to do with the plant, but a similar set of steps. One of the challenges in plants is that plants have much more complicated genomes, they have sexual reproduction and so forth, and so one of the first steps in a plant is trying to understand how much variation exists in the plant genome, and that's something that's much more computationally intensive than what we would do, say, in a microbe. And the next one is something in communities, and again, just skip through the animation here. One of the challenges in communities is that the vast majority of organisms in the environment or in your gut or anywhere, we can't actually culture. And usually to study an organism, we need to culture it to produce a pure culture from which we can then extract the DNA, from which we can then use all these tools. But we want to study the 99% of the organisms, the 99% that we can't grow. And one strategy for doing that is to directly sequence a community, sequence a metagenome, and to assemble that genome, that is to build from the small reads to assemble it into large fragments and then sort it based on some kind of binning algorithm by separating the genome fragments that belong to different organisms into clusters that should correspond to a single organism, and then invoking the modeling pipeline I'm talking about to predict things about those individual organisms. And one of the things that we could predict then is what the growth media needs to be to enable us to grow a particular organism. And we've done this on several organisms already, and we've built the tools now to apply this at large scale. So hopefully this kind of technology would help us break this bottleneck of getting access to the 99% of the organisms. The next slide talks about how you might integrate all of this, say, to build models of, say, soil ecology. I think in interest of time, I'm going to skip through it. I can just say that soil is very complex. A typical gram of soil will have about 10 to the 9th organisms, and it might have in excess of 10,000 different kinds of organisms. But not all of those organisms would exist at the same abundance. And most of the chemical activity in the soil is probably the result of only a handful, maybe 20 or 30 different kinds of organisms. Since we can attack this with sequencing, it becomes possible to imagine building models of something as complex as soil and be able to predict the chemistry that a community is able to undergo in soil. Okay, so go, again, I don't know what slide number it is, but you want to go to the slide that says at the top what KBase need to provide. So if you haven't tuned out yet on this, you can realize that this is a very complex undertaking with lots of tools, complex data, integration, models, and what really does the environment need to provide to enable productivity? Well, first of all, in order for it to be worthwhile to build an infrastructure, one needs to provide to a user a capability they don't have. And what that means in the context of computational biology is access to a scalable compute environment that's dramatically larger than what they have access to locally, and data handling capabilities that are beyond what we can have locally. And there are different strategies for doing this. Some projects like iPlant are really attacking the direct infrastructure challenge. Something like KBase is not doing that. We have an infrastructure, but we're trying to build mostly on the tools and provide tool-level access, which behind the scenes is going to exercise this infrastructure. So there's different ways of doing this. We need a distributed infrastructure that's available 24/7 worldwide. It has to be integratable with local systems, meaning that there's ways of seamlessly or almost seamlessly uploading and downloading data to the system and maintaining security and so forth. And the whole idea here is to make this very easy to use so that biologists can focus on the biology and the computer scientists and technology people can make the infrastructure work. Next slide. This is the cartoon slide. You may have seen this slide before. You've all probably flown, and nowadays you get to choose where you sit in the airplane, sort of. And this cartoon just sort of indicates our philosophy that our real vision here is not to tell users what they're able to do with the infrastructure, but to make it easy for them to be in control of what they want to do.